Hello, today we're going to talk about radiobiology research and why they matter for space exploration. I want to acknowledge Leonard Fisk and Tom Hay for extending me the invitation and my funding support from the National Cancer Institute and University of Nevada in Las Vegas. So our argument is that the uncertainties in the projection model are the major issue, not that the risk is higher than lower than what's acceptable. And it's really the radiobiology that needs to be addressed and improved knowledge and data sets, not more information about environments or shielding, which for designing a mission are reasonably well known. So safety requirements are a lifetime risk limit. So the US, we have the National Research Council and NCRP providing guidance. We have the 3% limit. And because of the uncertainties, we have a 95% confidence level. Other limits should be on during mission, avoid radiation sickness and cognitive detriments. And then other late effects would be possibly limited. The limits and acceptable levels of risk determine mission and vehicle requirements for space radiation protection. And we also have the ALARA principle. So the design environments are well known. This is a model we published fitting different data sets, including the more recent Voyager 1, which help us understand the modulation better. Very high quality fits are found for the GCR, and this shows you the solar modulation. The chi-square per degree of freedom are fairly well understood in reproducing the data. Solar particle events, we know over 400 events since 1950. And looking at the largest 43, we see that a reasonable amount of shielding material would reduce it to a very low probability of having an excess of the 30-day limit for the blood forming organ or for any possibility of acute radiation sickness. So the issue seems to be alert dosimetry and make sure the crew get to the shielding. The predictions are reasonably well for integral quantities, 10% for dose and dose equivalent. This is the model we use compared to the RAD data. People are excited about new measurements in space, Mars or the Earth's moon, but we predicted these things fairly well a long time ago. So I prefer to say these new data sets validate what was known rather than giving us new information. So from all this information, we know that shielding is not playing a major role for GCR. Solar minimum, you have some decrease in effective doses or dose equivalent for small amounts of shielding, but as you lose all the low energy particles, it becomes very flat. And for solar max, you don't even have the reduction for small amounts because of the absence of the lower energy particles or reduction in their number. So the issue is the risk and how can we predict these? It's a different type of radiation than Earth. So using X-rays or gamma rays to predict what will happen in space is not conclusive. People always say, how many equivalent X-rays was that space exposure, but it's apples and oranges. You can't make that comparison. So one issue was cataracts. And this one's illustrative because from X-rays and gamma rays, we expected a threshold dose above, say, 1 gray, but we saw an increased risk for 8 millisieverts. And for a much longer, higher dose mission like the Mars mission, this caused some concern that the cataracts would occur during the mission. This was plot here on the right is for what we call non-trace cataracts for visual impairment and was involved. And these older data sets were reproduced and enlightened by state-of-the-art measurements by Leo Shalak of the Brigham Woods Women's Hospital and later publications. So this is interesting because it shows using X-rays was wrong. It didn't help us understand this risk. Not interesting so much because it shows our lens dose limits for space missions are being violated, at least there are 10 of them, but because these things are very easily cured post-mission as a minor surgery, but it shows we were wrong and the possibility of occurring during a long mission like a Mars mission of 30 months or more, and it might have implications for other late effects. So CNS risk are one area I want to talk about in more detail. So this is a risk that was driven by the early observation of light flashes, and there was some information in studies with heavy ions looking at loss of neuronal cells, but that really didn't show too much. And then more recently, that it's taken off with more extensive cognitive testing of animals. But the main issue for me is the idea of a dose threshold. So CNS risk would be characterized as deterministic or non-stochastic, so quite different than cancer risk, which are stochastic. So there should be a dose threshold, and the severity of the risk increases above the dose threshold, which is different than cancer risk, which the risk is the same, it's just the probability is going up with dose. So this becomes the issue, is to study this risk and understand the thresholds. And to a large extent, what's been done is at doses well above what we would expect on a Mars mission, at least the heavy ion dose. So this is caused by, the concern is caused by studies of neurogenesis and persistent oxidative damage, and then all along, and then more recently, more extensively, cognitive tests. Remind you, cognitive tests can focus on different areas of the brain and their function. One area that's been studied is the hippocampus, and the hippocampus is a good area for, in the sense that mice and rats have a similar structure and function as the hippocampus in humans, while other areas are less developed than in humans, for example, the prefrontal cortex. So these studies are kind of mixed at the lower doses. Certainly half a gray or higher, they seem to show a positive or detrimental effect, and but not at the lower doses. Some investigators see effects and others do not. There's no studies in larger animals, and then there's some question about genetic strain and things like that. That are still open questions, and how you translate it to humans. So here's one that's the most widely used, called a novel object recognition memory. This is how a slide from, the pictures from Bernie Rabin at University of Maryland. So what they do is they train an animal to see if an object, and they call that the familiar object, and then after irradiation and in the control group, they introduce a novel object, and they measure the time it takes for, the animal spends exploring the objects. This tests various regions of the brain, medial temporal lobe and parental cortex, with some role for the hippocampus. And what he sees is at higher doses, there's an effect with iron particles, but here, for example, no significant effect at 10 rads, actually a slight increase in performance. So we took a meta-analysis approach looking at Bernie, but everyone's data, about 60 publications. We defined a familiar to novel ratio, and then a relative risk. And then we looked at the relative risk. And for the Mars mission, we're interested in this dose rate against 0. So I could say it's less than 0.1 gray, about 0.05 gray would be the cumulative heavy ion dose. And we fitted data and then made predictions. This was published in Life Science and Space Research last year. And you can see there's a mild risk, but with the uncertainty in the model and the data, it certainly could be no risk or slightly higher risk than we predict. One thing interesting is this NOR test is also used in mouse models of Alzheimer's disease and Parkinson's disease, also traumatic brain injury. So we can make a comparison, and you can see the Mars mission prediction is slightly less than these disease model predictions. Another test is this for spatial memory. This is done in different ways in labs. So we also made a relative risk model. And here the meta-analysis suggests there's no effect at lower doses. This white graph here shows you the heavy ion doses for different components. That's an average solar cycle. And you can see it's quite low for individual charge groups, and the cumulative, like I said, is less than 0.1 gray. So when we're down in this region, it's inconclusive whether there's a risk. So next we turn to the mechanisms. So there's a variety of mechanisms that are involved and have been proposed and studied. Mostly these are correlative, that people aren't looking at multiple mechanisms in the same model. So if some of the older studies by John Fike and others looked at impaired neurogenesis and correlated that with cognitive changes, but they didn't look at the other one. So you don't have a bona fide proof that was causing it. Neuron morphology, neuroinflammation goes across all these, the role of support cells. So we looked at the neurogenesis. Mind you, this is the one area of the brain where you continue to make new neurons throughout life. They have chemical approaches to label the cells, and then they have double labeling to look at proliferative cells, and then they can look at time after exposure and see if the number of cells and the proliferation of the cells is changing with dose and different types of radiation. So we have a nonlinear model that we use to make some extrapolations of the data sets. What we found is we can predict a threshold dose for impaired neurogenesis at a long time point. This would be 9 months after irradiation. And we find, and this is consistent with some of the later talks by John Fike, that this would not be an issue below about half a gray, maybe a little bit lower, but we're only interested below 0.1 gray for the cumulative heavy ion dose. So it doesn't seem to be an issue. Another issue that altered neurogenesis is genetics. And most of the studies were with 57 black 6 mice, largely male, and a few studies with female. And with different rat strains exposed to X-rays, you get a large divergence of responses. So we don't know how this would line up with heavy ions for these different rat strains, but also which strain, which animal model would best represent humans. The other is the mouse hippocampus. There's some schematics of the axons, the neurons, and different layers of the hippocampus. And then each neuron has a soma, the cell nucleus, and then the dendritic tree where the spines and the synapses are formed. So the University of California, Irvine has published a series of publications looking at irradiating mouse hippocampus with fluorescent markers of the spines, and they can also measure the length of the dendrite. So they look at a plane of cells, maybe 100 neurons are involved, but they don't have an exact count. And then they measure the changes in the length or the number of spines, and they see changes at fairly low doses for acute radiation for several types of heavy ions. One of the problems with this assay is they don't know how many cells were there before and after the radiation. So we did some modeling, and we looked at radiation tracks in hippocampus, and then looked at the detailed damage to individual neurons. And we made some predictions, this one for titanium. And we found this problem of not knowing how many cells can influence your conclusion. That so for single cells, this would be the damage. For the population, this would be the damage. So the population curve is because of that factor. You don't know how many cells were there. Okay, so the summary of the CNS risk would be that I don't think countermeasure research is called for at this point, that the risk is not established to the extent where we don't know if it's really plausible. So you might have a study of and spend a lot of money on countermeasures for a risk that doesn't exist, that there's relevant dose and mixture of particles in space. And we don't know, I'm aware of underreporting of negative results, that's common, but also in this area, how to extrapolate from mouse to humans, hippocampus, maybe the other areas more problematic of the brain, genetic studies. I showed you the rat impaired neurogenesis study, and then combined stressors in space from space radiation, microgravity, sleep deprivation, other things. So I would think that the resources are better spent looking at low doses below 5 rads, 0.05 gray, and how to extrapolate results to humans. So turning to cancer risk, this is something that most people look at cancer as being caused by radiation, radiation acting as an initiator, causing mutations and chromosomal rearrangements, leading to cancer decades later. New research says there's important roles for the tissue microenvironment and immune responses. This is a schematic from Marianne Barcelo-Hoff. She has a nice chimera model to prove those points. We developed a model in 2012 that was reviewed by the National Research Council and the NCRP, and then also in 2015 by a NASA external board called RCAP. So we were able to integrate all the data using track structured based theories of radiation quality, and we did the uncertainty analysis in very lot of detail, introduced a new model of based on BEER7 to convert from incidence to mortality. The problem here was epidemiology data. When mortality does not take into account the effect that cure rates and differences between countries occurred. So when applying the Japanese A-bomb survivor data to US population, we felt it was better to use the incidence data. Also a never smoker data that lowers the risk for astronauts compared to a US average population. we've made some predictions. This is for individual cancers, total cancer incidence and cancer death, and then the probability causation that radiation was the culprit if a cancer was observed. So these are problematic, some of them are exceeding 50%. So you would have a conclusion that it was caused by radiation if it did occur in an astronaut. The uncertainty in these curves is mostly driven by the large range of RBEs that we use to determine quality factors in the mouse solid tumors. This gives you an example, all the data. This is the strength of the quality factor function that we use. And you can see it almost 2 orders of magnitude with all the data. If we exclude liver, which has been studied in male mice and a very high RBE, hard uterine gland, which is a common model because it's easily accessible and a shorter time period to do the study. It's about an order of magnitude. So, but that order of magnitude is still large, and that's the biggest driver of the uncertainty. The other thing is this conventional model and the NSCR model before 2015 was only the qualitative differences, the quantitative difference. We want to look at qualitative differences as well. And here we see a different shape of the dose response curve indicating a qualitative difference, an earlier appearance of tumors with heavy ions compared to gamma rays, and a different spectrum of tumors. So these are all things that are not taken into account in conventional approaches. The life loss is the bottom line for the cancer risk. So 3%, but what 3%, but the detriment is how much life will be lost. So leukemia, 23, and most of the solid cancers about 15 years, and for heart disease, about 9 years. A little bit different between males and the females also have breast and ovarian cancer risk to worry about. So if the time to tumor is shorter at high LAT, these numbers would go up. And so you would judge the detriment to be much higher. That's a concern. Metastases. So Bob Ulrich has shown with liver cancers in male mice that metastases is much higher with heavy ions, and a similar result by Al Forney. He's not looking at metastases, but invasive carcinoma versus other types of colorectal cancers, and also looking at intestinal cancers in transgenic mice. So qualitative difference here. So when we use this data to guide some estimates, you can see the uncertainty would grow appreciably if you have these qualitative differences. So you can read our past publications in this to give the details. Then the other major area is non-targeted effects. So these are models this is an approach that says that it's not just DNA damage and mutation, it's also damaged cells alter the biochemistry of the surrounding tissue. And they go by labels of bystander effects, genomic instability, tissue microenvironment changes, and less important for NASA or space travel would be abscopal effects, which is certainly important in therapy. This alters the shape of the dose response. So if it's a flat response, maybe mission length doesn't matter, maybe shielding matters less. So you certainly want to know these non-targeted effects. One example was by Edison. He used very low doses of alpha particles. And from the dose and the size of the cell, you can estimate how many cells have been traversed by an alpha particle. And in this study, it's about 1%, but he sees a signal of a DNA damage responding protein involved in cell cycle controls, P21, in many more than 1%. And then he looked at cells that are deficient in gap junction communication, and then the effect goes away. Other studies along the similar lines use ROS inhibitors to show the effect goes away. Something's going on where the damaged cells are communicating, and it's in the dose region we are interested in. Another way to look at this is look at a dose response, linear here, something going on at the lower doses. Take this data and divide it by the x-axis, so divide the transformation by the dose, and then change it to fluence rather than fluence normalized by the area of the cell. And you can see that at one, if I took this data and it was linear and I divided by the dose, it should be a constant line. And when I do this, it's a constant line at the higher doses, but at the lower doses, you have this rapid increase. So that's another indication of a non-targeted effect. And this is the region we're interested in. We're not interested in this higher dose region. And same things with chromosome aberration from past study I did with Megumi Hata and Kerry George. So conventional model would vastly underpredict what's happening at the space relevant doses for a Mars mission. And then it increases our prediction using those data sets to modify the quality factor with a non-targeted effect component, we see that now the cancer death prediction can go up above 20% for females and approach 15% for males. So what might be relax the 3% limit, if you're going up around 8%, becomes very problematic if you're approaching 20% to do that. Last issue I want to point out is the chronic inflammation. So space station astronauts, it's been shown that when there's some immune system changes in space, but it goes back to baseline a few months after the mission. But if you have a longer time period, 6 months versus 3 years, and about an 8 times higher organ dose on the Mars mission, that combination of chronic inflammation with continued damage to cells would suggest that there's another component to the risk that we're not taking into account. So I think this also should be a large focus. This falls into line with what Mary Helen Barcelahoff is suggesting. So summary, if the conventional models use, we can say that it's 8 or 9%, under 10% at the 95% confidence level will be the fatal cancer risk. But if it's incorrect, we're approaching a 20%. So I think this is not allowable, not problematic. This perhaps some people would like to keep it to 3%, but maybe they relax it for an important mission like going to Mars. This one becomes more, if this is correct, it's more like playing Russian roulette with the crew. So altered immune response could increase this even higher. And so along with the potential role of cognition during the mission, I think these would be the major issues. Thank you.